Hi everyone, welcome to MR Learning Circle. I hope everyone is fit and fine and doing good in their lives. So this is the 26th lecture of your microbiology MCQ series, and in this lecture we are going to cover the entire topic, entire unit of your microbial genetics. And if you have not watched the previous videos, you can find the link above here, or you can find the link in the description box, as well as if you want to get the PDF or the ebook of your microbiology notes, MCQs, as well as multiple select questions, you can find the link to get them in the description box. So now let's start with our topic, that is microbial genetics. And the first question for here is, which mutation most likely results from UV irradiation? Options are A, GC to AT transition, B, AT to GC transition, C, formation of thymine dimers, D, frame shift mutation. So the correct answer for this question is option C, that is formation of thymine dimers. Now question number 2. Which chemical mutagen causes alkalization of O6 of guanine? Options are A, nitrous acid, B, hydroxylamine, C, ethyl methane sulfonate, D, acridine orange. So the correct answer for this question is option C, ethyl methane sulfonate, ethyl methane sulfonate, that is EMS. Question number 3. Acridine dyes primarily causes, options are A, transition, B, transversion, C, frame shift mutation, D, silent mutation. So the correct answer for this question is option C, frame shift mutation. Question number 4. The base analog that pairs with both A and G during replication is, options are A, 5-bromouracil, B, EMS, C, nitrous acid, D, proflavin. So the correct answer for this question is option A, that is 5-bromouracil. Question number 5. Which mutation is least likely to alter protein functions? Options are A, nonsense, B, missense, C, silent, D, frame shift. The correct answer for this question is option C, silent. Now question number 6. Nitrous acid causes mutation by, options are A, alkylation of bases, B, deamination of bases, C, interaction between bases, D, cross-linking DNA strands. So the correct answer for this question is option B, deamination of bases. Question number 7. Deamination of cytosine produces, options are A, thymine, B, uracil, C, hypoxanthin, D, xanthin. Correct answer for this question is option B, uracil. Question number 8. Which repair system directly reverses UV damage without excisions? Options are A, base excision repair, B, nucleotide excision repair, C, photoreactivation, D, mismatch repair. So the correct answer for this question is option C, photoreactivation. Question number 9. Replica plating is mainly used to identify. Options are A, nutritional mutants, B, conditional lethal mutants, C, antibiotic resistant mutant, D, auxotrophs. So the correct answer for this question is option D, auxotrophs. Question number 10. In AIMS test, mutation reversion is detected in strains of, options are E. coli TRP minus, Salmonella typhimurium histone minus, Bacillus subtilis leucine minus, Pseudomonas aeruginosa methionine minus or negative. Option is Salmonella typhimurium histone negative. Question number 11. The role of liver extract in AIMS test is to, options are A, repair DNA damage, B, increase mutation rate, C, activate pro-mutagen, D, neutralize toxin. So the correct answer for this question is option C, activate pro-mutagen. Question number 12. A chemical showing mutagenicity only in presence of X9 mix is, options are A, direct mutagen, B. Base analog, C. Pro-mutagen, D. Anti-mutagen. So the correct answer for this question is option C. Pro-mutagen. Question number 13. Transformation involves uptake of. Options are A. Single-stranded DNA, B. Double-stranded DNA, C. RNA, D. Plasmid only. Correct answer for this question is option B. Double-stranded DNA. Question number 14. Competence in bacillus is regulated by. Options are A. SOS response, B. quorum sensing, C. heat shock protein, D. plasmid genes. So the correct answer for this question is option B. quorum sensing. Question number 15. In conjugation, DNA transfer begins at. Options are ORI C, option B ORI V, option C ORI TA, or option D TER. The correct answer for this question is option C, that is ORI T. Question number 16. An HFR strain is found when. Options are A. Plasmid is lost, B. F plasmid integrates into host chromosome, C. chromosome integrates into plasmid, D. 2 plasmid recombine. So the correct answer for this question is option B. B plasmid integrates into chromosome. Question number 17. Which recombination requires Rec A protein? Options are A. Site-specific recombination, B. Homologous recombination, C. Transposition, D. Illegitimate recombination. So the correct answer for this question is option B. Homologous recombination. Question number 18. Generalized transduction occur during. Options are A. Lysogeny, B. Lytic cycle, C. prophase induction, D. specialized excision. So the correct answer for this question is option B. Lytic cycle. Question number 19. Specialized transduction transfers gene. Options are A. randomly from chromosome, B. adjacent to the prophage insertion site, C. only plasmid gene, D. from donor cytoplasm. Correct answer for this question is option B. adjacent to prophage insertion site. Question number 20. Which phase is classic example of generalized transduction? Options are A. lambda, B. T4, C. P1, D. M13. So the correct answer for this question is option C. P1. Now question number 21. R plasmid confer resistance by gene located on. Options are A. IS element, B. operon, C. transposon, D. promoter. So the correct answer for this question is option C. transposons. Question number 22. Composite transposons are characterized by. Options are 1 IS element, B. 2 IS element flanking genes, C. no inverted repeats, D. site-specific recombination. So the correct answer for this question is option B. 2 IS elements flanking genes. Question number 23. Which enzyme is required for transposition? Options are A. integrase, B. recombinase, C. transposase, D. ligase. Correct answer for this question is option C. transposase. Question number 24. Replicated transposition produces A. loss of transposons, B. 1 copy only, C. 2 copies of transposons, D. no target duplication. So the correct answer for this question is option C. 2 copies of transposons. Question number 25. Mut-S, Mut-L, Mut-H are involved in. Options are A. photoreactivation, B. base excision repair, C. mismatch repair, D. SOS repair. So the correct answer for this question is option C. mismatch repair. Question number 26. Which repair mechanism is error prone? Options are A. base excision repair, B. nucleotide excision repair, C. photoreactivation, D. SOS repair. So the correct answer for this question is option D. SOS repair. Question number 27. DNA glycosylase act during. Options are A. mismatch repair, B. base excision repair, C. nucleotide excision repair, D. recombination repair. Correct answer for this question is option B. base excision repair. Question number 28. A repressor binding to operator prevents. Options are A. RNA polymerase binding, B. translation, C. DNA replication, D. termination. So the correct answer for this question is option A. RNA polymerase binding. Question number 29. Inducible operons are usually involved in. Options are A. Biosynthesis, B. Housekeeping, C. Catabolism, D. DNA repair. Correct answer for this question is option C. Catabolism. Question 30. The lac operon is. Options are A. Constitutive, B. Repressible, C. Inducible, D. Attenuated. So the correct answer for this question is option C, that is inducible. Question 31. Allolactose act as A. Repressor, B. Inducer, C. Co-repressor, D. Attenuator. Correct answer for this question is option B. Inducer. Question number 32. CAMP and CAP complex binds near promoter to. Options are A. Inhibit transcription, B. Enhance RNA polymerase binding, C. Degrade RNA, mRNA, D. Block operator. Correct answer for this question is option B. Enhance RNA polymerase binding. Question number 33. The trip operon is regulated by A. Induction only, B. Repression only, C. Attenuation only, D. Repression and attenuation. So the correct answer for this question is option D. Repression and attenuation. Question number 34. Attenuation depends on. Options are A. Ribosome stalling, B. DNA methylation, C. RNA splicing, D. Protein degradation. Correct answer for this question is option A. Ribosome stalling. Now question number 35. The E. coli genome is approximately. Options are A. 1 MB, B. 2 MB, C. 4.6 MB, D. 10 MB. So the correct answer for this question is option C. 4.6 MB. Question 36. Genes in E. coli are arranged mainly as. Options are A. Interons, B. Operons, C. Exons, D. Isochores. The correct answer for this question is option B. Operons. Question 37. E. coli chromosome replication initiates at. Options are ORI T, ORI V, ORI C, TERC C. So the correct answer for this question is ORI C. Question number 38. The lambda phage attachment site on E. coli is. Options are A. LAC-Z, B. GAL, C. AT-B, D. ORI-C. Correct answer for this question is option C. AT-B. Question 39. Decision between lytic and lysogenic cycles is regulated by. Options are A. CRO and CL protein, B. REC-A and LEX-A, C. CAP and CAMP, D. sigma factors. So the correct answer for this question is option A. CRO and CL protein. 40. Question number 40. Lambda prophage integrates via. Options are A. Homologous recombination, B. Transposition, C. Site-specific recombination, D. Illegitimate recombination. So the correct answer for this question is option C. Site-specific recombination. Question 41. RNA viruses have higher mutation rates because. Options are A. Short genome, B. No proofreading, C. Segmented genome, D. Host polymerase. So correct answer for this question is option B. No proofreading. Question number 42. Resortment occurs only in virus with. Options are A. 42. Double-stranded DNA, B. Single-stranded DNA, C. Segmented genome, D. Circular genome. So the correct answer for this question is option C. Single-stranded DNA. Question number 43. Antigenic shift is due to. Options are A. Point mutation, B. Recombination, C. Reassortment, D. Deletion. Correct answer for this question is option C. Reassortment. Question number 44. Antigenic drift results from. Options are A. Frame shift, B. Point mutation, C. Resortment, D. Integration. So the correct answer for this question is option B. Point mutation. Question number 45. The core genome represents. Options are A. All genes in a species, B. Genes present in all strain, C. strain specific genes, D plasmid genes. So the correct answer for this question is option B, genes present in all strains. Question number 46. Pan-genome includes, options are A, only conserved genes, B, only accessory genes, C, core plus accessory gene, D, viral genes. So the correct answer for this question is option C, core plus accessory genes. Question number 47. Comparative genomics helps in studying. Options are mutation rate only, B, gene regulation only, C, evolution and pathogenicity, D, protein folding. Correct answer for this question is evolution and pathogenicity. Question number 48. Which mutation restores wild type phenotype by compensating another mutation? Options are A, forward mutation, B, back mutation, C, suppressor mutation, D, conditional mutation. Correct answer for this question is suppressor mutation. Question number 49. A temperature sensitive mutant is an example of A, conditional lethal mutant, B, auxotrophs, C, prototrophs, and D, revertant. So the correct answer for this question is option A, conditional lethal mutant. Question 50. Which system increases mutation rate under stress? Options are A, photo repair, B, base excision repair, C, SOS response, D, mismatch repair. So the correct answer for this question is option C, SOS response. 51. F plus into F minus conjugation results in, options are A, both HFR, B, both F plus, C, F minus, D, HFR plus F minus. So the correct answer for this question is option B, both F plus. 52. Abortive transduction results in, options are A, stable recombinant, B, partial diploid, C, transient gene expression, D, cell lysis. Correct answer for this question is option C, transient gene expression. 53. Plasmid with narrow host range replicate in, options are A, many genera, B, only one species, C, gram positive only, D, viruses. So the correct answer for this question is only one species. 54. Which DNA repair removes bulky lesions? Options are A, base excision, B, mismatch, C, nucleotide excision, and D, photoreactivation. Correct answer for this question is option C, nucleotide excision. Question 55. Operator mutation preventing repressor binding leads to A, no transcription, B, inducible expression, C, constitutive expression, and D, attenuation. So the correct answer for this question is C, constitutive expression. Question number 56. Which RNA act as regulatory molecule in attenuation? Options are A, rRNA, B, tRNA, C, leader mRNA, D, miRNA. So the correct answer for this question is option C, leader mRNA. 57. In lambda phase, CI protein promotes, options are A, lytic growth, B, lysogeny, C, DNA degradation, D, host lysis. So the correct answer for this question is option B, lysogeny. Question 58. Viral recombination requires, options are A, host ribosome, B, co-infection, C, reverse transcriptase, D, segmented genome. Correct answer for this question is option B, co-infection. Question 59. Which genome has highest gene density? Options are A, eukaryotic, B, archaeal, C, bacterial, D, viral. Correct answer for this question is option D, viral. Question 60. Horizontal gene transfer includes, options are A, transformation, B, transduction, C, conjugation, D, binary fission. So the correct answer for this question is option D, binary fission. Now statement based question. Question 61. Which statements about transposons are correct? Statement 1. causes insertional mutagenesis. B, require Rec A. Third statement is have inverted repeats. So options are only 1. Option B 1 and 3. Option C 2 and 3. Option D 1, 2 and 3. So the correct answer for this question is option B 1 and 3. Question number 62. Correct about lac operon. Statement 1, glucose inhibits transcription. Statement 2, allolactose inactivates repressor. Statement 3, cap binds operator. Now the options are 1 and 2. Option B is 2 only. Option C is 1, 2 and 3. And option D is 1 only. So the correct answer for this question is option A, 1 and 2. Now question number 63. Which is true for generalized transduction? A, require specific gene transfer. B, require prophages. C, any gene can be transferred. D, no phage involved. So the correct answer for this question is option C, any gene can be transferred. Question number 64. Viral mutation rate is highest in. Options are A, double-stranded DNA virus. B, single-stranded DNA virus. C, RNA virus. D, retrovirus. So the correct answer for this question is option C, RNA virus. Now question number 65. Genome islands usually indicate. Options are A, vertical inheritance. B, mutation hotspot. C, horizontal gene transfer. D, gene silencing. So the correct answer for this question is option C, horizontal gene transfer. Question number 66. Which gene is not part of lac operon? Options are A, lac Z. B, lac Y. C, lac A. D, lac I. So the correct answer for this question is option D, lac I. Question number 67. The amirodiploid is formed during. Options are A, transformation. B, HFR conjugation. C, abortive transduction. And D, lysogeny. Correct answer for this question is HFR conjugation. Question number 68. Prophage induction is triggered by. Options are A, nutrition limitation. B, UV radiation. C, antibiotics. D, temperature drop. So the correct answer for this question is option B, UV radiation. Question number 69. Which process contributes most to bacterial evolution? Options are A, mutation. B, recombination. C, horizontal gene transfer. D, selection. So the correct answer for this question is option C, horizontal gene transfer. Question number 70. The main aim of microbial genomics is to. Options are A, study morphology. B, identify pathogens. C, understand function and evolution of genes. D, culture microbes. So the correct answer for this question is option C, understand function and evolution of genes. So that was all for this lecture. I hope you have learned something from this video. If yes, then please do like, share and comment down below. And this PDF would be available on the Telegram channel. You can go to the Telegram channel. The link of the Telegram channel is mentioned in the description box, and you can get the PDF of this particular topic in the Telegram channel. Thank you for watching. Keep learning.